Swastiastu. Good morning, sir, and good morning, everyone. Today, I want to present about carbohydrate metabolism too. Before that, let me introduce myself. My name is Putumera Chandrachehani with license 1011. So the first question about citric acid cycle or Krebs cycle. Explain the role of the citric acid cycle in energy metabolism and its relationship with the end product of glycolysis. The citric acid cycle acts as the center of energy metabolism in the mitochondria, which oxidize acetyl CoA into CO2 or carbon dioxide. In this process, NADH or FADH2 and GTP or ATP are produced, which are then used in the electron transport chain to generate a large amount of ATP. Its relationship with glycolysis is that the end product of glycolysis in the form of pyruvate will be converted into acetyl CoA, which then enters the Krebs cycle. Thus, the Krebs cycle is a continuation of glycolysis under aerobic conditions. Uh, the second question about analysis of cellular energy disorder. Based on the case, the patient experienced chronic fatigue. Analysis the relationship between the disruption of the Krebs cycle and decreased energy or ATP production. Disruption in the Krebs cycle causes a decrease in the production of NADH and FADH2. These two molecules are very important in the electron transport chain to produce ATP. If the amount of NADH and FADH2 decreases, then ATP production also decreases. As a result, cells experience a lack of energy, which is characterized by chronic fatigue. In addition, the body tends to switch to anaerobic metabolism, which produces lactic acid, so the level of lactate in the blood increases. Next question about regulation of gluconeogenesis. Explain the mechanism of regulation of gluconeogenesis and the factors that influence it, or hormones and cellular energy. So, gluconeogenesis is regulated by hormones and cellular energy status. The hormones glucagon and cortisol stimulate gluconeogenesis, while insulin inhibits it. In terms of energy, low ATP condition will stimulate gluconeogenesis, while high ATP will inhibit it. In addition, regulation also occurs through K enzymes such as pyruvate carboxylase, PAP carbokinase, and fructose 1,6-biphosphatase. Thus, gluconeogenesis is active when the body lacks glucose, such as during fasting. Next question, role of the Cori cycle. The patient shows an increase in lactate levels in the blood. Explain the role of the Cori cycle in this condition and how the body overcomes it. The Cori cycle functions to connect metabolism within muscle and the liver. In muscles, glucose is converted into lactate through anaerobic glycolysis. Lactate is transported to the liver and converted back into glucose through gluconeogenesis. In this case, the increase in lactate indicates that anaerobic metabolism increases. However, because liver function is impaired, the conversion of lactate into glucose is not optimal, so lactate accumulates in the blood. Next, about pentose phosphate pathway or PPP. Explain the main function of the pentose phosphate pathway in cells and its relation to the cellular needs for NADPH, and biosynthesis. The pentose phosphate pathway functions to produce NADPH and ribose 5-phosphate. NADPH is used in biosynthetic reactions such as fatty acid synthesis and as an antioxidant to protect cells from free radicals. Meanwhile, ribose 5-phosphate is used as a component of nucleotide formation in DNA and RNA synthesis. Thus, this pathway is very important for biosynthesis needs and cell protection. Next question. Analysis of the pentose phosphate pathway. Why is the pentose phosphate pathway important in tissues such as the livers and adipose tissue? Analyze its relationship with lipid metabolism. So, the pentose phosphate pathway is important in the liver and adipose tissue because these tissues are active in fatty acid and cholesterol synthesis. This process requires an ADPH as a source of electrons. An ADPH produced from the pentose phosphate pathway is used in the lipogenesis process. Therefore, this pathway plays an important role in supporting lipid metabolism, especially in fat formation. Next question. Glycogen synthesis or glycogenesis. Explain the process of glycogen synthesis from glucose and the physiological condition when this process occurs. Glycogen synthesis begins from glucose, which is converted into glucose 6-phosphate, then into glucose 1-phosphate, and subsequently into UDP glucose before finally being assembled into glycogen by the enzyme glycogen synthase. This process occurs when blood glucose levels are high, such as after eating, and it's stimulated by the hormone insulin. Its purpose is to store excess glucose as energy reserves. Next question. Glycogen degradation or glycogenolysis. Explain the process of glycogen degradation and how this process helps maintain blood glucose levels. Glucogenolysis is the process of breaking down glycogen into glucose 1-phosphate by the enzyme glycogen phosphorylase, which is then converted into glucose 6-phosphate. In the liver, glucose 6-phosphate can be converted into free glucose and released into the bloodstream. This process is important to maintain stable blood glucose levels, especially during fasting or when the body needs energy. Next question. Regulation of glycogen metabolism. Analyze the role of the hormones insulin and glucagon in regulating glycogen metabolism. Insulin plays a role in lowering blood glucose levels by stimulating glycogenesis and inhibiting glycogenolysis. In contrast, glucagon functions to increase blood glucose levels by stimulating glycogenolysis and inhibiting glycogenesis. These two hormones work antagonistically to maintain the balance of blood glucose levels in the body. The last question, integration of carbohydrate metabolism. Based on the case above, explain how the Krebs cycle, gluconeogenesis, pentose phosphate pathway, and glycogen metabolism are interconnected in maintaining the body energy balance. Carbohydrate metabolism is an interconnected system. Glycolysis produces pyruvate which enter the Krebs cycles to produce energy. When oxygen is limited, pyruvate is converted into lactate, which then enters the Cori cycle and is converted back into glucose through gluconeogenesis. Glucose can be stored as glycogen through glycogenesis and broken down again through glycogenolysis when needed. Meanwhile, the pentose phosphate pathway provides an ADPH for biosynthesis and cell protection. In the patient case, Disruption in several of these pathways cause decreases energy production, increases lactate, and imbalance in blood glucose levels, so the body is unable to maintain energy homeostasis optimally. Alright, that's all for my presentation, and thank you.